Hi everyone, this is video part 2 of our lecture 18 here on photosynthesis and the Kelvin cycle. So this will be our last chapter of our course this year. And I want now to build on what we had discussed in our part 1 video on photosynthesis and the light reactions and apply what we had learned there to the Kelvin cycle and actually look at how we can make sugars through the Kelvin cycle. So this video will really focus on our third learning goal here. We want to be able to explain the processes of the Kelvin cycle, how it is connected to the light reactions of photosynthesis, and how is it relevant for the biosynthesis of glucose and other metabolites. So just to recap, right, in the first video in part 1 of our lecture, we had talked about the photosynthetic light reactions and how we can use this light-driven ETC to produce ATP in our electron carrier here, NADPH. So what do we do now? How can we use these in our dark reactions in the Kelvin cycle to make glucose? All right, so now we're on the right part here. We had talked about our light reactions in the first video. Now we're here. We want to use the NADPH and the ATP we just generated in the Kelvin cycle here, where we incorporate gaseous carbon dioxide. We're using the energy from ATP and the energy from our electrons in NADPH to bring about the formation of sugars. That's our job here. So in general, right, what the Kelvin cycle is, it is essentially fixation of gaseous carbon dioxide, and we're using our ATP and NADPH from the light reactions to bring this about. So what we can do, we can use this energy, we're taking this gaseous carbon dioxide and we're reducing it essentially down to the level of glyceraldehyde 3-phosphate, our G3P, right, our glycolytic and GNG intermediate, which is our smallest carbohydrate. And that's what we can use to build all other sugars, and that's our food. Right? So if you put this concept in numbers, right, what you're getting here is that you're taking on 3 carbon dioxide from the air, you're using the energy of 9 of your ATPs and 6 of your NADPHs that you generated in the light reactions, and you're converting these 3 carbon dioxides into 1 3-carbon carbohydrate G3P, and you're using the energy from your ATP and NADPH. So it's a pretty expensive process, but if you consider that you can take carbon dioxide from the atmosphere and you can turn it in a sugar, that seems to be a pretty worthwhile investment, right? So the people who actually identified the Kelvin cycle and elucidated how it works were Melvin Kelvin. He was a professor at UC Berkeley at the time, and some of the researchers in his group, Andrew Benson and James Bassam, those 3 were really the key people who elucidated how the Kelvin cycle actually works. As a laboratory head, Melvin Kelvin received the Nobel Prize. The Kelvin cycle was named after him. Right. But then later, seeing that they didn't get a Nobel Prize for it, but they contributed largely to this work, gradually the name of the Kelvin cycle was changed, first to the Kelvin-Benson cycle, then to the Kelvin-Benson-Bassam or CBB cycle. And it's also called the C3 cycle because you're generating a 3-carbon carbohydrate. And we'll see in a little bit why it's actually also called the reductive pentose phosphate pathway. All right. So the way these guys actually figured it out is that they used a single-celled green algae called chlorella. It's actually, you can buy this also as a supplement, for example, for health benefits. But it's a green algae and has an advantage that it has very large chloroplasts. So they're really good for experimental purposes. And so they use this study system to try and understand how the Kelvin cycle in the chloroplast actually works. And so they set up a pretty smart experiment that's highlighted here. It's actually called the green algae lollipop experiment. And so what they did is they developed just a glass platform here. Right here, it's just a thin piece of glass. and it has sort of the stem down here that is facing into hot methanol. And so inside here, you have your green algae, and you feed in radio-labeled carbon dioxide, right? We're talking about fixating carbon dioxide, so you can radio-label this, you can feed it into these algae. And so they did this, they kept them in the dark, and then they have a light source here, just a lamp. And so they can turn on the light into the system, and then they have a timer here, and they can basically now release the green algae from the system at any point in time, right? So you have your algae in here, you turn on the light, and after whatever time you wish, you release this, and the algae are flowing into the hot methanol, and the hot methanol will extract all the metabolites from the algae. Right? Having done this, right, what you do now, you take the metabolites in your hot methanol, and you can do a thin layer chromatography. Nowadays, you would do metabolomics, so mass spectral type of approaches. Back then, you would use thin layer chromatography or TLC. What you basically do is you put your compounds here on this sort of base layer on, for example, a paper or another matrix that can be used for this. And you have your solvent down here. And what is happening now is that the solvent will gradually move up the paper and will carry these metabolites with it. And depending on the chemical and physical properties of these metabolites, they will travel at different rates on this plate, and you can separate them thereby. And because you used radio-labeled carbon dioxide, you can expose this with a radio film, and you can then see on this plate here all the metabolites showing up as black that are radio-labeled. Right? And then you can do this for all these metabolites that you know. You can do the same, but not with your experiment, just with this compound. So you can basically use these compounds as standards and identify what all these compounds are. Right? And that's exactly what they did. And it's intriguing what they found, right? If you see here, this is now a TLC after 5 seconds of light exposure with these green algae that have been fed with radio-labeled carbon dioxide. And you see after 5 seconds already, right, you have your PGA, you have your PAP, you already have the first sugar phosphates here. So even after 5 seconds, you're already well into your gluconeogenesis. Right? If you wait a little longer, 30 seconds, now the algae already have made sucrose, they have made amino acids here, they have made TCA cycle intermediates here. And it's amazing to see how fast metabolism actually operates, right? Even after a few seconds, you already have made amino acids, right? If you think about how many lectures it took us, right, to come all the way to sugars and then potentially from sugars through the TCA cycle to amino acids, this is all done in just a few seconds. And this experiment now can be used, right? Sort of, if you do this at different time points, you see sort of at 5 seconds, you have these metabolites, 10 seconds, you have these. You can understand what the series of reactions actually is, right? Which metabolite is built after the other. And so you can understand the series of reactions in a metabolic pathway using this fairly simple but really ingenious system. And that's exactly what people have done. Right, now that we understand like how Melvin Kelvin and his team have figured it out, we want to start looking at what are actually the reactions that are important here. Okay. And so before I start, I want to sort of highlight that we will actually see a lot of reactions that you have already encountered this quarter. So there are actually only 2 new reactions that we will have to learn today. Everything else you actually already have seen. And there will be reactions of the pentose phosphate pathway and GNG. So if you don't quite remember how GNG and the PPP worked, I would encourage you to maybe pause here and have a look back at that material first, because I think it will really help you to understand the Kelvin cycle, to just remind yourselves of what the reaction were of the PPP and GNG and glycolysis. But the first enzyme here is actually one that we haven't seen before. That's a new one. It's called the ribulose 1,5 bisphosphate carboxylase oxygenase. So that's a mouthful, of course. So you will know it, and most people call it simply Rubisco. Rubisco is actually a very large protein complex. It's divided up into 2 different subunits, each of which carries 8 large and 8 small subunits. So it's a big enzyme complex that's just highlighted here. It actually turns out that Rubisco is the most abundant protein in plants. And given how much plant biomass we have on the planet, it is actually the most abundant protein on Earth. And part of the reason for its abundance is that it is catalytically extremely slow. It's really a bad catalyst in terms of speed. And so you need a lot of the protein molecules to be able to push this pathway efficiently. In part, this has to do with also other functions that Rubisco has that we won't touch on here in this lecture, that you have basically a compromise between catalytic speed and multiple possible functions that it can be used for. But as a result of it being slow in the direction we will look at now, you need a lot of this protein. All right, what does it do? So it's actually a reaction we haven't seen yet. We have seen parts of it, right? So what it does actually, it takes ribulose 1,5-bisphosphate. That already should ring a bell, right? We had talked about ribulose 5-phosphate in our PPP, right? That was one of our 5 carbon sugars. So you're taking a 5 carbon sugar here in form of a biphosphate in this case. Right? And then you go through actually 2 different reaction steps. And so they're highlighted here in brackets, just these are the enzyme-bound intermediates, right? They will never be released from the enzyme. But you're taking your 5 carbon biphosphate. And the first thing you do is basically an isomerization here of this carbonyl group. So you're moving the double bond from the carbonyl group over to the carbon-carbon bond here, right? And now you have an alkene and you have a hydroxy group, you're generating an enol. Enol should ring a bell, right? We had talked about enols in the context of enolic phosphates. So if you would just have another phosphoryl group here, it would be an enolic phosphate. That was a high energy compound. Now the next step you do is you're taking this double bond here, which is now more reactive than the original carbon-carbon single bond. You can modify it, and what you're doing is actually you're carboxylating it. So you're bringing carbon dioxide in here. This is the enzymatic step of the carbon fixation, catalyzed by Rubisco, and you're bringing it in here at C2. Okay. So you're attaching it. This is one of the other examples of enzymes that can transfer carboxy group independent of our biotin cofactor. Right? So this happens independently. So now you have this carboxy group sitting here, right? It's sort of within the enzyme. You now have 6 carbons here, but you started with a 5 carbon sugar. You bring in your 1 carbon as carbon dioxide. Now you have a 6 carbon intermediate. And what you can do now is cleave it. And you can use water to do so. Because, right, you have a carbonyl group here. So the oxygen is drawing away electrons from this carbon. You have a carboxy group here and a hydroxy group. So there are also electrons drawn away from this. So you're really weakening this bond here. And you bring in water and you can cleave it into 2 3-carbon molecules. And because of this original isomerization and the carboxylation at this position specifically, you're actually cleaving them into 2 identical molecules, and that's 3-PGA, right? Our glycolytic and GNG intermediate. So what you're doing, right, you're cleaving here. That leaves you with this carboxy group. Then the second carbon has the hydroxy group, and up here the phosphoryl group. And down here, this carbonyl group will take on the second oxygen from the water in this hydrolysis process. So you're actually incorporating that oxygen molecule. It's making it a water-dependent oxygenation. You're generating this carboxy group here, and then the rest is just the bottom end here of your enzyme intermediate. So Rubisco, what it does, it can fixate carbon dioxide from the atmosphere, can bring it into a 5-carbon intermediate, generate a 6-carbon intermediate, and then split it into 2 3-carbons. Fairly efficient, interesting way of generating and incorporating additional surplus carbon. All right. Now that we have done this, we want to go further, right, with the cycle. So we haven't yet done a cycle. So going into the cycle, just as a reminder again, right, you're bringing in ribulose 1,5-bisphosphate. You do carbon fixation with your Rubisco. You're generating 3-PGA. And you're actually doing so 3 times. So similar to the PPP, we have to do these cycles multiple times now in order to be able to recycle our substrate here, which is ribulose 1,5-bisphosphate. So keep in mind, in order for the Kelvin cycle to operate, we need to run Rubisco 3 times, okay? So we're getting 6 molecules of 3 PGA. Okay. All right. Then the next reaction, right? We've just generated your 3 PGA right here. The next step is reduction, okay? So what we're actually doing is now, we're doing 2 things. We're using actually our ATP from the light reaction, and we first phosphorylate the carboxy group here, right up here. You bring the phosphoryl group in here, then you make 1,3-bis PGA, right? That was another GNG and glycolytic intermediate. And then you're using your NADPH here from the light reactions. It's being oxidized. That tells you already what's going to happen here, right? You're reducing, and you're reducing this group here now, right? You have activated it with phosphorylation. Now you're reducing it down to an aldehyde, and that's G3P. Okay. So this should ring a bell if you sort of look at these reactions, what that looks like, right? If you were to go back from G3P, oxidation combined with phosphorylation all the way back to 3-PGA here, that was glycolysis, right? The other way around, 3-PGA, 1,3-PGA to G3P, that's GNG. Okay. So we're helping ourselves here simply by using the same reactions as GNG, but it turns out that these are actually isozymes of the gluconeogenesis that we already had talked about that are specific to the chloroplast. So these are isozymes catalyzing the same reaction, but unlike the GNG, these are located in the chloroplast. Okay. All right, now we have made G3P, that's great. We have our 3-carbon intermediate. And now it becomes clear why we did this 3 times in the beginning, right? What we want to do is that we want to generate G3P, we want to generate sugars, and we want to regenerate our substrate, right? So if you actually look at the carbon balance, right, what we have generated is 6 molecules of G3P, a 3-carbon intermediate that makes 18 carbons. Right? We now actually need 5 of those to regenerate 3 of these ribulose 1,5-bis phosphate substrates for Rubisco. Right? Keep in mind that's a 5-carbon carbohydrate, so you only need 15 carbons, which means that the 3 carbons that you have incorporated through fixation of carbon dioxide, this is now the G3P that is actually can be used for the production of your glucose, for production of other carbohydrates, amino acids, etc., etc. So you have used now carbon dioxide fixation to bring in additional carbon that you now can use for any purpose of anabolic pathways to generate a variety of different biomolecules. But you're bringing in a lot more carbon for the purpose of being able to run this as a cycle and to regenerate your 5-carbon precursor for Rubisco. Right? If you wouldn't do this, there would really be little point because you would otherwise have to make your ribulose 1,5-bis phosphate somehow. So recycling really makes it an efficient cycle of generating new carbohydrate precursors. All right, so we have made our 1 G3P that we want to use for biosynthesis. We have made 5 that we want to recycle. So let's have a look at how this recycling actually works through the Kelvin cycle. So here's the whole thing. Again, don't worry. You have seen the majority of the reactions already as part of your GNG and PPP lectures. But keep in mind what we want to do now is, right, we want to recycle 3 of our ribulose 1,5-bis phosphate molecules. And we do so bringing in 5 of our G3Ps. There's an error here. This should be G3P. We're using those to regenerate them. Okay. So I'm going to highlight step by step now how this actually works for us. Right? So I've highlighted the numbers here. So we're using this G3P intermediates here. And 1 by 1, 4 different reactions. So it's not just a cycle. There are also multiple different reactions that are happening. Okay. So let's have a look at how this works. I'm just going to switch to the pen here. And so first I'm going to do like this actually should be G3P. So that we know this. And so, okay, step by step, how this actually can work. So step 1 is we want to use our 5 G3Ps here, right? We will convert 2 to DHAP, right? This is part of GNG again. We can use an isomerization to interconvert DHAP and our G3P. And so one of the G3Ps we're bringing in here now, and we're bringing in one of the DHAPs. We're doing an aldol condensation of these 2,3 carbon intermediates, right? What we get is a 6 carbon intermediate, right? 1, 2, 3, 4, 5, 6 carbons. We have phosphoryl groups at the C1 and the C6. We have our keto function here. And then if you look at the positions of your hydroxy groups here, left, right, right, you should be able to actually recognize what this is, right? That's actually fructose 1,6 bisphosphate, right? Again, another GNG reaction going from our 3 carbon intermediates now through aldol condensation. We're generating fructose 1,6 bisphosphate. We can use our phosphatase, right, to bypass the irreversible kinase reaction of glycolysis. So we're using the reverse reaction, the phosphatase, another GNG reaction. We are getting to our fructose 6-phosphate. Okay, so these are all GNG reactions. Then next up, what we're doing next are we're now looking at, we're hijacking some of our pentose phosphate reactions to further convert this, right? What we want to do is we want to have 5 carbon sugars. What we have generated here in fructose 6-phosphate is a 6 carbon sugar. So what we do next is we're bringing in our fructose 6-phosphate right here. Right? And we're bringing in another molecule of our G3P, right here. And what we're generating now is your xylulose 5-phosphate, a 5 carbon, and your erythrose 4-phosphate, a 4 carbon. Right? That should ring a bell from your PPP reactions. So if you remember your non-oxidative part of the PPP, right, what we had done in this part was that we took xylulose 5-phosphate and erythrose 4-phosphate, and we used our transketolase reaction to convert this, right, through an aldol cleavage and aldol condensation type of reaction to generate fructose 6-phosphate and G3P. Now we're just using the same reaction in reverse, right? We're taking another molecule of G3P and we're taking the fructose 6-phosphate that we just generated and turning it into this 5 carbon xylulose and the 4 carbon erythrose intermediate. Okay. But we want all 5 carbons, so we're not done yet. So the next step, right? What we're doing now is we're taking our erythrose 4-phosphate that we just generated, and we're taking basically another DHAP here that we just generated from another molecule of G3P, right? There's G3P right here. That can be isomerized. So we're taking another one of this. And so we're bringing those together. That's actually our other new reaction here. I will not ask you to really look at it or know it for the exam. Just recognize it's a different kind of reaction that we hadn't seen yet. That's essentially an aldol condensation as we have done it in the PPP, but it works mechanistically a little bit differently. The outcome is the same as in the PPP. What we're generating is, right, a C7 sugar. This is our pseudohaptulose 1,7 bisphosphate, right? So it's a biphosphate in this case because we are actually bringing in 2 phosphates, erythrose phosphate and DHAP, right? So that's the difference to what we have seen in the PPP. But what we can do is we can use another phosphatase here, right? You're dephosphorylating your bisphosphate at carbon 1. You're now coming to your PPP intermediate here, your pseudohaptulose 7-phosphate. Okay. Moving on, right? Still, we are not at all 5 carbon sugars yet. Now what you're doing is that you are taking this pseudohaptulose intermediate that we just generated here. Oh, apologies. Pseudohaptulose that you generated here, and you're bringing in your last of the 5 G3Ps. So all the way down here, you bring them together. And what you're generating with those, that's another pentose phosphate pathway reaction. You're now generating another molecule of xylulose 5-phosphate, and you're generating another molecule of ribose 5-phosphate, okay? So again, that's your PPP, right? These isomerization reactions that we could do between ribulose and ribose and between ribulose and xylulose. So have a look at those. And if you go back, that's exactly what we did. We did this reaction of converting a C7 and a 3C intermediate to 2 5-carbon intermediates. So we have all 5 carbons now, right? We can now, we have one molecule of ribose 5-phosphate here. We have one molecule of xylulose. Right? If you look at this now, right, you can use isomerization to convert xylulose to ribulose, and you can now convert ribulose to ribose. So we're good. We can make all ribose 5-phosphate. So that's fine. But right, what we want is ribulose 5-phosphate and not ribose. So we just did this under conversion. Now that we have all ribulose, what we want ultimately is ribulose 1,5-bisphosphate. So we need another phosphoryl group, and we can do this through a kinase reaction. We can use the remaining ATP of your light reaction to phosphorylate your ribulose 1,5-phosphate to ribulose 1,5-bisphosphate, and that's really your last reaction. We now have recycled successfully the substrate for Rubisco, and so this recycled ribulose 1,5-bisphosphate can now undergo another reaction of carbon fixation. So that's the last reaction. That's how we can use all of our ATP and our NADPH to generate new sugars in form of G3P. That can be used for all sorts of anabolic reaction, and in this entire Kelvin cycle, using parts of GNG and parts of our pentose phosphate pathway, we can regenerate again, as we have done for all our other cycles, the secondary substrate, so we can have a very energy efficient and carbon efficient way of doing the formation of sugars using ultimately light energy. And so this is it. This was the last reaction. Congratulations, we have come to the end. You are a BIS103 finisher. Job well done. I hope, even though we couldn't actually go through all this material together and have a bit more of an interactive way of talking about these pathways, that these videos and our office hours that we'll hold online will be helpful and will end up still being a valuable and interesting experience to sort of dive into how metabolism actually works, all its intricate cooperations and interconnections, and how it's really fundamental to sustaining life. And so I'll leave you with the last pathway here. You can meander over this, and I'll hopefully see you in the office hours over the quarter now as it is starting. And I wish you all the best, and hopefully it will be successful.